All right, so real quick, all I want to talk about in regards to microscopy were the 3 cell types that you looked at. I think everything else I kind of cover in my review videos on the website. I talk about slide orientation. If I want to cover that real quick before I show you the cells, remember you guys looked at an E under the microscope and it didn't look like this. It was inverted. Okay? Real quick, there's a super fast trick you can always remember and always do, and it will always show you what a letter looks like under the microscope. All you do is take a piece of paper and you write the letter the way that you normally see it, and then you turn the piece of paper completely upside down. And whatever you see, that's how the letter looks under the microscope. So if I turn this paper like that, that E is going to look like that. Super easy. My students, if you can't remember, I should see you turning your papers if they ask you that question. Okay. All right, now, let's talk about the 3 different cell types. I just want to talk about the structures that you guys were looking at in those cells. Now, I'm not going to use any green for the Elodia because it shows up poorly on the board, so you're just going to have to use your imaginations. All right? So, we looked at 2 different plant cells underneath the microscope. The first cell type that we looked at came from the organism Elodia, which is a photosynthetic plant. Okay. Now remember, for each of these cells, we had to use a specific stain to help us visualize things inside the cell. Anybody remember what the stain was we used with Elodia? Iodine. Right? It was iodine. And when we used that iodine, we saw a variety of structures inside the Elodia cell. We saw these photosynthetic organelles, okay, which are, of course, called chloroplasts. We also saw the outermost structure of the cell, which is the cell wall, because it's a plant cell. And then if we were lucky, we might have actually stained that central structure that is indicative of any eukaryotic cell, which of course is the nucleus. Now if you didn't see the nucleus, that's okay, because that's why we did the second plant cell type. So these were the big things we were going for with this particular plant cell. Now we did a second plant cell as well, which we got from an onion. And again, what stain did we use here? Iodine. We used iodine yet again. Okay. And when we did that, what you hopefully saw was this kind of elongated cell with a very, very dark structure in the center. And inside that structure, you might have seen a couple of different circles. Now again, these are plant cells, so what's my outermost structure called? The cell wall. Okay, and then that centralized structure there, the nucleus again. And then these circles, in theory, are nucleoli, or the nucleolus, which is the primary manufacturer in the nucleus of ribosomal RNA. Now, why didn't I see chloroplasts in my onion? Because the onion is a bulb that grows underground. So it's not going to have chloroplasts because that part of the plant doesn't perform photosynthesis. It's the stalk that does. The stalk of the onion, like the green part, that's the part that performs photosynthesis, not the bulb. Now, outside of plant cells, we also did animal cells, right? Okay, and what animal in particular did we focus on? Humans, specifically. Yes, specifically your nasty cheeks. I'm just kidding. We did use cheek cells, and what was our stain that we used? Methylene blue. Good. Trick question, what was the color of this stain? Blue. Good. If only that were a question on the practical, huh? Alright, so these cells looked more like this. Might be a little difficult to see. They didn't have a nice rigid structure. Instead, they kind of have this globular mass. And that's because what's the outermost structure on an animal cell? Plasma membrane. Good. That central structure on the inside of the cells was the nucleus. Good. And then the region that exists between the plasma membrane and the nucleus is called the cytoplasm. Excellent. that's really all I have to say on microscopy. Now, outside of that, I would say knowing the different parts of the microscope and the relative function is always a good idea. And I've got pictures with descriptions, I believe, on the website. Also, knowing how to view things under the microscope can't hurt you. And I think I also go through the chart about the depth of focus and all of that. Okay? So watch those review videos.